Hello everyone. The topic we are going to discuss is the Krebs cycle. Now you might have also heard this called the TCA cycle or the citric acid cycle. All three names refer to the same pathway. This is one of the most important topics in biochemistry. So let us go through it together step by step. I am Brinda Ronika, tutor working at M College of Nursing. Before we start, let me just quickly tell you what we are going to cover. So first we'll define what the Krebs cycle is, then we'll see where it takes place in the body. After that we'll go through all the steps one by one with the enzymes, and finally we'll see how much energy this cycle produces and why it's so significant. Okay, so let us begin. What exactly is the Krebs cycle? The Krebs cycle or TCA cycle is basically the process by which our body takes a molecule called acetyl coenzyme A and completely breaks it down into CO2 and water. Now you might be wondering, okay, but why is that important? See, when we eat food, whether it is rice, chapati, oil or protein, all of that food gets broken down, and eventually, no matter what type of food it is, it all gets converted into one small molecule called acetyl CoA. Carbohydrates becomes acetyl CoA, fats become acetyl CoA, even proteins after breaking down becomes acetyl CoA. So the Krebs cycle is a common final pathway where all of this comes together and gets oxidized to release energy. That's why this cycle is called the central metabolic pathway. It is literally at the center of everything happening in the body metabolism, and it does not just produce energy, it also produces intermediates, small molecules that are used to make amino acids, glucose, heme in our red blood cells, and so many other things. So in short, the Krebs cycle is one of the most important things happening inside our cells every single second. Now let us look at the formal definition. The Krebs cycle is defined as the oxidation of acetyl CoA to CO2 and H2O. Simple, right? Let us just break it down quickly. Acetyl CoA is the starting molecule. It has two carbons. Oxidation means electrons are being removed from it. When those electrons are captured by molecules like NAD plus and FAD. So NAD means nicotinamide adenine dinucleotide and FAD means that is flavin adenine dinucleotide. They carry the energy to the next stage, the electron transport chain, where the ATP is produced. The two carbons from acetyl CoA are eventually released as carbon dioxide, that is literally the CO2 we breathe out every time we exhale, and water is also formed as a byproduct. So food and acetyl CoA Krebs cycle, CO2 breathed out, water and energy captured. That is a big picture of this Krebs cycle. Okay, so now where does this cycle actually take place? In humans and in all eukaryotes, the Krebs cycle takes place inside the mitochondria. More specifically in the mitochondrial matrix, that is the innermost fluid-filled space inside the mitochondria. This is why the mitochondria is called the powerhouse of the cell, because that is literally where energy production happens. Now in prokaryotes like bacteria, since there are no mitochondria, the cycle runs in the cytoplasm instead. And one more thing, there is a connection to glycolysis here. Glycolysis happens in the cytoplasm and produces pyruvate. That pyruvate enters the mitochondria, gets converted to acetyl CoA and then enters the Krebs cycle. So the flow is glycolysis in the cytoplasm produces pyruvate. Pyruvate enters the mitochondria, becomes acetyl CoA, and then the Krebs cycle runs inside the mitochondria. Alright, now we are getting into the most important part, the steps of the Krebs cycle. There are 8 steps in total. Let us go through them one by one. Step 1 is the formation of citrate. The cycle starts when acetyl-CoA, which is a 2-carbon molecule, combined with oxaloacetate, which is a 4-carbon molecule. When these two join together, they form citrate, which is a 6-carbon molecule. The enzyme that does this job is called citrate synthase. Now, oxaloacetate is a very important molecule. It is a molecule that welcomes the acetyl-CoA into the cycle, and here is the interesting thing. At the very end of the cycle, oxaloacetate is regenerated again. So it keeps coming back ready to receive the next acetyl-CoA. That is what makes this a cycle. So to say it simply, acetyl-CoA 2 carbons plus oxaloacetate 4 carbons gives us citrate 6 carbons, and the enzyme is citrate synthase. Step 2 is the isomerization of citrate to isocitrate. Now we have citrate, but here is the thing, citrate cannot be directly oxidized. It needs to be rearranged first. So the enzyme aconitase comes in and does a two-step rearrangement. First, it removes a water molecule from the citrate to make cis-aconitate. This is just a short-lived intermediate. Then it adds a water molecule back to the form of isocitrate. So the molecule has the same atoms. The formula has not changed, but the arrangement is now different. This is called isomerization. And now isocitrate is in the perfect shape for the next step. So to say it simply, citrate becomes cis-agonitate and then becomes isocitrate. And the enzyme doing both steps is agonitase. Next, step 3 is the oxidative decarboxylation of isocitrate. This is where things get exciting. This is the first time energy is captured in the cycle. The enzyme here is isocitrate dehydrogenase, and this step happens in 3 sub-steps. First, isocitrate gets oxidized, meaning a hydrogen is removed and transferred to NAD+, converting it to NADH. This NADH will go on to make ATP later. Second, a CO2 molecule is released. This is the first carbon dioxide to leave the cycle. Third, the remaining molecule rearranges itself into alpha ketoglutarate, a 5-carbon molecule. A cofactor called manganese helps facilitate this reaction. So after step 3, we have gone from 6 carbons down to 5 carbons. We have produced 1 NADH and we have released 1 CO2. The enzyme is isocitrate dehydrogenase. Next, the step 4 is the oxidative decarboxylation of alpha ketoglutarate. Now, if you look at this step, it looks very similar to step 3. And you are right, it is another oxidative decarboxylation. The enzyme is the alpha ketoglutarate dehydrogenase complex, a large multi-enzyme machine. Alpha ketoglutarate, which has 5 carbons, gets converted into succinyl-CoA, which has 4 carbons. In this process, the second CO2 of the cycle is released. So both carbons from the original acetyl-CoA have now been removed. Another NADH is produced and a CoA group is attached to the product, forming succinyl-CoA. These steps need vitamin B1, B2 and lipoic acid to work properly. So vitamin deficiency can actually affect this cycle. To say it simply, alpha ketoglutarate gives us a succinyl-CoA plus carbon dioxide plus NADH, and the enzyme is the alpha ketoglutarate dehydrogenase complex. Next, step 5 is a conversion of succinyl-CoA to succinate. Now here is something special about this step. The enzyme is succinyl-CoA synthetase and it converts succinyl-CoA into succinate. What makes this step special means This is the only step in the entire Krebs cycle where a high energy molecule is produced directly in the form of GTP. GTP, which stands for guanosine triphosphate. GTP is basically the same as ATP. Same energy value, and it's produced directly here without going through the electron transport chain first. GDP plus inorganic phosphate gives GTP here, and CoA is released as a byproduct. To say it simply, succinyl-CoA gives us succinate plus GTP, and the enzyme is succinyl-CoA synthetase. Next, step 6 is the dehydrogenation of succinate to fumarate. The enzyme here is succinate dehydrogenase, and it converts to succinate into fumarate. Now, in most steps we have seen NAD plus picking up the electrons, but in this step the electron acceptor is FAD, not NAD plus. So FAD gets reduced to FADH2. Why does it matter? Because FADH2 carries slightly less energy. It produces only 2 ATP when processed by the electron transport chain compared to NADH, which gives 3 ATP. There is also something very unique about succinate dehydrogenase. It is the only Krebs cycle enzyme that is embedded directly into the inner mitochondrial membrane. All other enzymes float freely in the matrix. This one is physically attached to the membrane. So actually what is happening here means succinate gives us fumarate plus FADH2, and the enzyme is succinate dehydrogenase. Next, step 7 is a hydration of fumarate to malate. This is a simple step, just the addition of water. The enzyme fumarase adds an H2O molecule across the double bond of fumarate to produce L-malate. The L here is important. It specifically produces L form of malate, which is a biologically active form. The enzyme is very specific about this. So no NADH, no FADH2, no GTP here, just simple hydration reaction. So to say it simply, fumarate plus water gives us L-malate, and the enzyme is fumarase. And the next step, last step, is a dehydrogenation of L-malate to oxaloacetate, and this is a final step of the Krebs cycle. The enzyme is malate dehydrogenase, and it oxidizes L-malate to produce oxaloacetate. In this step, one more NAD plus is reduced to NADH plus H plus, and the product oxaloacetate is the exact same molecule we started with back in the step 1. So now the cycle is complete. This fresh oxaloacetate is ready to grab another acetyl-CoA and run the whole cycle again. This is the beauty of a cycle. It never really ends long as acetyl-CoA keeps coming in. To say it simply, L-malate gives us oxaloacetate plus NADH, and the enzyme is malate dehydrogenase, and the cycle begins again. So now look at this diagram. This shows a complete Krebs cycle with all 8 steps together. Let us chase it quickly. Oxaloacetate is at the top. Acetyl-CoA enters and joins it, and citric acid is formed by citrate synthesis. Moving clockwise, citric acid becomes isocitric acid through aconitase. Then isocitric acid becomes alpha-ketoglutric acid through isocitrate dehydrogenase, releasing CO2 and NADH. Then alpha-ketoglutric acid becomes succinyl-CoA through alpha-ketoglutarate dehydrogenase, releasing another CO2 and NADH. Then succinyl-CoA becomes succinate through succinyl-CoA synthetase, producing GTP. Then succinate becomes fumarate through succinyl dehydrogenase, producing FADH2. Then fumarate becomes malate through fumarase using water, and finally malate becomes oxaloacetate through malate dehydrogenase producing NADH. And we are back to the start. The cycle is complete. Please practice drawing this diagram with all the enzyme names. It is one of the most commonly asked diagrams in your biochemistry exams. So now let us talk about the most important part. How much energy does the Krebs cycle gives us? Remember, the cycle runs 2 times for every one glucose molecule. Why? Because one glucose breaks into 2 pyruvate, which gives 2 acetyl-CoA, which means 2 turns of the cycle. So here is the energy from 2 complete turns. From the 2 isocitrate to alpha ketoglutarate reactions, we get 2 NADH and that gives us 6 ATP. From the 2 alpha ketoglutarate to succinyl-CoA reactions, we again get 2 NADH gives us another 6 ATP. From the 2 succinyl-CoA to succinate reactions, we get 2 GTP directly, which is 2 ATP. From the 2 succinate to fumarate reactions, we get 2 FADH2 gives us 4 ATP. And from the 2 malate to oxaloacetate reactions, we get 2 more NADH giving us 6 ATP. So, if we add all of that, 6 plus 6 plus 2 plus 4 plus 6, we get a total of 24 ATP from the Krebs cycle. This energy does not come directly from the cycle itself. The NADH and FADH2 carry the energy to the electron transport chain where the most of the ATP is finally made. So the Krebs cycle is like a charging station. It charges up the NADH and FADH2 carries, which then go and make ATP. So that is the Krebs cycle. All 8 steps, all the enzymes, the site, the definition and the energy yield of 24 ATP. To quickly recap, the Krebs cycle is the oxidation of acetyl-CoA to CO2 and water. It takes place in the mitochondrial matrix. It has 8 steps starting and ending with oxaloacetate. It produces 3 NADH, 1 FADH2 and 1 GTP per single turn and a total of 24 ATP for 2 turns. And it is the central hub connecting all metabolic pathways in the body. Thank you.